was extremely long, as I said at the beginning of this symposium today, but in fact we are missing one very important keyword, one of David's main passions, which was RNA, because David, as you know, had a deep interest in RNA biology, stability, structure, etc., and we're going to hear more about that certainly from Simon and other people today. So I'm absolutely delighted to welcome Trudi Schupbach, also from Princeton University, who will introduce that theme for us today and talk about David in Basel, early discussions with David on RNA localization. Thank you, Trudi.
Good. Oh, I need to go to the beginning. Yeah. Okay, and to the presenter mode. Good. And I'll use this. Well, thank you, Nick. I see jumping to that slide. Sorry, somehow it jumps to the last slide. That one. So, first, thank you. Yes, the mic. Thank you, Roz. Thank you to the organizer, and also hello to everybody, all old colleagues and friends here. I'm very honored to be able to talk to you today about about David, even though it's a sad occasion, but it's nice to be celebrating a wonderful person. I met David already when he was in Basel. Now, I was a graduate student. Sorry. I was a graduate student at the time in Zurich, and of course, David was in Basel in the Bierzentrum, but Switzerland is small, and we were exchanging people right and left. We went to Swiss Drosophila conferences together, so I did meet David at the time. And in fact, Walter Gehring and my thesis advisor, Rolf Nötziger, were graduate students together in the laboratory of Hadorn some time ago, together with another Drosophila scientist whom you maybe know, Gerald Schubiger. So all three of them were together graduate students in the same lab. And so there were lots of connections here. We PhD students did our thesis work actually here in the upper parts of the Zoological Institute under the roof, whereas the postdocs like Eric and Janos Sabad were in the bigger lab, nicer lab downstairs. But nevertheless, we had a lot of fun here. You see that we could actually climb out of these windows and sit on the roof, which we sometimes did. I did not really scientifically interact with David at the time because, as Eric pointed out, they were working on heat shock and we were working on things like sex determination and development of the genital disc and the ovary and important things. But I remember spending some time with David after Eric came to Zurich, obviously. In Hungary, there was a Drosophila conference, which Mike Ashburner was there, and Peter Lawrence, and then us students. And after the conference was over, Eric and I and David stayed a day extra, and we went to a concert in Budapest. Then David visited in Heidelberg, Germany, where he at one point helped with a little bit with the screen. We were trying to sort out embryos, dead embryos, and David was much better than, for instance, I was in doing that. Later, after David moved to London, we visited him in that house that he had with this magically overgrown garden, which maybe some of you remember. And then he moved to Oxford and we to Princeton. And again, I remember we visited him, and now jump a little later, 93 in Oxford, where he took us, we visited with our kids, and he took us to this museum, the Ashmolean, and I'm just bringing this up because it was clear. I'm not sure who was more enthusiastic, our girls or David, about all these bones that one could look at and drawers to open and see fantastic things. So 93 was though the time that David and I started to talk seriously about science, because and exchanging ideas, because at that time a postdoc in my lab, Shira Neumann-Silberberg, had cloned and therefore molecularly started to identify the Gurken gene, which I had been working on for quite some time, because it was an important gene that was involved in setting up the dorsoventral polarity of the egg and embryo of Drosophila. So what you're seeing here is an egg chamber, and for non-scientists or non-Drosophila people, so this will eventually give rise to the egg, a mature egg, that the female will then lay. It will be fertilized and give rise to the embryo. And interestingly, or very excitingly, we found that the Gurken RNA gets localized or accumulates to high levels in this very defined region within this whole egg chamber. It overlays the oocyte nucleus, which is right under here. So this is the RNA, and then leads to the production of localized protein that you can see here. Now, this picture reminded, of course, David, or something that he and Elon Davis and other people in his lab had looked at, and that is the localization of parallel transcripts in the embryo. So they found and published that in this particular picture, FUTS transcripts, which is expressed in 7 stripes, are localizing apically above the embryonic nuclei, which are not visible here, but are lying right underneath here. And so this compared again to, if you look here, we have the nucleus, and again we have transcript localized in very close proximity to the nucleus. So we were talking about what mechanism could do that. Well, how could that? Could there be a unified mechanism that keeps these transcripts close to the nucleus, or what's going on? It became more concrete when, after Amanda Norwell, another postdoc in my lab, found that a particular gene which is called SQUID in flies, led to that mutant. When this gene was absent, then the Gurken RNA wasn't so nicely localized to just this corner above the oocyte nucleus, but now it was found all along the length of the anterior end of the oocyte, leading again also then to mislocalized protein. Now, SQUID encodes an HNRNP protein, and these proteins are very conserved. It's a whole family of proteins. They are called heterogeneous nuclear RNA binding proteins, because if you stain for those, you find them accumulating inside nuclei. At the time, nobody kind of was really sure what they were doing. They were supposed to be maybe involved in splicing. But here we found that this particular HNRNP protein was obviously important in keeping Gurken, or bringing Gurken, or whatever the mechanism was, to this localized position. And so David said, maybe SQUID is also involved in having these parallel transcripts localized to the apical side. And even though that was a typical David idea, so it's something that you say, well, I don't know. And then he said, well, we should try it. Who knows? Because HNRNP proteins are conserved widely in organisms. Maybe that's their role. And so in my lab, then Amanda, who was good at doing some biochemistry, first did some cross-linking experiments where she labeled proteins, either from ovarian extract, labeled these with basically it labeled with P32 labeled either Gurken RNA FACS or nanos RNA, and found that using now an antibody to squid, found that and doing immunoprecipitation, found that squid can bind to Gurken RNA pretty well, can bind to FACS RNA, but does not seem to be very, have high affinity for nanos RNA. So then in David's lab, Sabi Lau did the injection experiments. So I need to first show some sort of control. Here you can see that they found, meanwhile, if they expressed just naked in vitro translated, in vitro transcribed FACS RNA, that it wouldn't localize. But if they pre-incubated with nuclear extract, it did localize to the apical site. So here, in another set of experiments, this was FACS RNA without its 3' UTR, and you can see the injected RNA just stays down here and does not move up to where the apical side of the plastoderm is, whereas after incubation, you can see it starts accumulating on the apical side. And so now, Amanda in my lab purified RNA from these different squid isoforms, and then we sent them to England. They took their RNA, incubated it, and injected it, and found that squid could promote this apical localization. Now, when I went back, of course, and I read the paper again, and I was surprised at how naive we were at the time and how little we knew. And we came up with sort of ideas, but again, in a way, the simplest idea was that, well, squid is already in the eggs, we know that, and maybe it just grabs onto these RNAs and transports them somehow up there. And of course, now we know we're much more sophisticated. We know that these HNRNP proteins have affinities that are not so specific. They actually can bind to many RNAs. We also can now understand what we did was actually we took a lot of RNA and a lot of protein and put them together. So obviously some of this binding protein will clog onto the RNA, but nevertheless, it obviously shows that something was happening. Was it that squid was then recruiting other proteins that were of importance, or did squid change maybe even the structure of the RNA in some way that then allowed the really important factors to bind? Is still open. But again, I just want to say that was very much David's pushing this idea and saying we should just go and try it, even though it's very risky. And here it worked out, and at least laid the ground for us to, from then on, have very more concrete ideas of what to do and how to test things. So this whole RNA field, and I think we will hear much more about it, started to explode around that time and led to us having RNA conferences, or wonderful conferences where RNA localization was discussed. Here is a picture of us in Colorado at such a conference, and you see here obviously Ilan and Liz Gavis and David. We did an excursion to a lake during the conference. We also all enjoyed very much these RNA conferences in Italy, in a place called Il Gioco, which is in Tuscany. And again, you can see David right here next to Liz, next to Anne Frussi. Ilan is here, and Daniel St. Johnston. The one in Tuscany was always a particular event also for me and David, because David would rent a car, and then one afternoon or so, he would drive down to Lucca in Tuscany, and he had already kind of investigated, of course, and knew exactly which restaurant we had to go to and have a very special Italian meal. He is with Eric, who once came to join us. We also enjoyed the Ringberg conferences, where, again, a lot of science was usually discussed over glasses of wine and beer, and then you could see David here happily participating. This was the last Ringberg conference that we were at, and I find David's expression here very typical of questioning Stefan about his work and not being quite convinced of, of the explanation. And then finally, the last time we were here in London, and David very proudly and very happily took us to the Crick Institute and showed us around and shared his enthusiasm for this new place with us. And I will also stop here and thank you for your attention.
Urgent questions for Trudi? Okay, if not, thank you very much, Trudi, for